Okay, I think the main idea is just to show how the EAD on the instruments works. And that's about a year that we have the instrument in the lab. Now I don't need to show you this anymore because it's already been introduced why it's important and where they are. Basically, they're in our blood. Apparently, a lot of immune responses of our body to environmental stress is actually motivated and controlled by the IgGs and IgAs. In this particular project that I'm about to show you, we've been using the IgA that had been derived from the kids who has allergies to peanut, and there is a kind of variety of the responses from the very light to very heavy, and apparently they've been separated, as well as of course control from the kids that do not have an allergy and enjoy peanut butter each morning. Those are assigned as a native, and then two other part of the cases would be something we call galactose and isolated. You will see further down the isolation part will be the most important, reaction to the allergy in this case. And, I don't want to tell you why it is important anymore. In this particular case setup, we had a ZenoTOF 7600 Plus with the EOSAP system up front of it. I really kind of enjoy to collaborate with the bioinformatics solution, and we've been using peaks all over this project, and mostly the peaks actually, because no other software can do this yet. That's why we've been introduced immediately after we install instrument in our lab, and I really enjoyed it, our collaboration. Thank you very much for the support, not only from SCIEX, but from the bioinformatics solution too. This is the instrument in our lab. We call it Harrison because we kind of go towards, building up a beetle in our lab. We already have linen. We already have the, as recently the year before, we came up with the Serpol. Now this is the Harrison. Harrison did pretty good job, I have to say, for the first year because we had a curve to get used to the software. Last time I've been using SCIEX about 15 years ago. That's why software slightly changed. Only slightly, but it did still change, and that's why I have to use it, to get used to it. Here, basically, the first interesting analysis that we have. We are fortunate enough, as I said, we have a Serpol in the lab, which is representing Astral, and we basically took the samples and split and start looking into the different analysis workflows. And as you can see, it's the same sample being analyzed by Astral weight workflow, ZenoTOF, and then we realized very quickly the triptych digest cleaves it too short for the analysis, and we would like to do something else. As already Albert was mentioning, that there is other necessity requires there is much longer peptides would be much better for the EAD, and we ended up with just using lyc for this particular case scenario. Lyc did work for us pretty well, and as you can see, we have very good coverage in this case scenario. We did do the reduction alkylation steps, and there is the whole IgE molecule is being represented here. Again, everything was analyzed by small database that we know about the IgE sequences, and it's been done by latest PEAKS 13. Thank you for providing us license for that. PEAK, if you look at the kind of in detail situation, we have a very small amount of the n-glycosylation side compared to the O-glycosylation side, and it was kind of quite interesting to see. And of course, the biggest drastic differences between the Astral data and the Xena data in both cases was the amount of the O-glycans that we picked. It was not surprising because clearly the EAD is the correct way of working with the O-glycosylation. And then respectively, the Astral, as you can see, there has much larger amount of non-glycosylated peptide covered compared to the instrument because, yes, it's created a lot of data, and that's not a surprising part too. And again, lyc gives us a little bit of boost on the coverage, a little bit an extra analysis of the peptide, and you will see later where it was very critical part of the story. It's not just by percentage only increase, but it actually was important for certain cases to see the coverage of the antibody, very critical pieces. This is how it's generally look like. Again, there is not any more news to you. You already have seen it from the previous talk. We have the ions, which is gives you ability to actually define what the peptide is from the C and Z ions, and then it's beautiful amount of the glycosylation peaks. I'm actually really still blown away by the amount of the intact, glycosylation ports we have here. It's been deja vu for me. I've been doing ECD about 25 years ago as a PhD student in Southern Denmark University, and it was a very different picture, I have to say. Now I'm back 25 years, different instruments, still the same problem, but intact, species. Was very interesting to see such a branch in sugars. That's another kind of area where you can show how the peak software define where the actually branch sugars are and why one another structure was assigned, as well as the clearly sequences where you can see the C and Z ions. I would like to pay attention to you in the bottom part. That's where the accuracy of the ZenoTOF data, usually within 5 ppm pretty much everywhere. Sometimes it's grow to 10 ppm, and the lower side where the Y1, Y2 ions, but further down it's actually very good accuracy for the fragments. Again, there's a data that has been recently published, and then our own data that we just obtained. It's not published yet, and you can see there has been already publication shows the regions where the typical N sites of the glycosylation being picked up, and how those look like. And you can see that I put a figure there from that publication. Please go ahead and use the publication if you need details from it. But our data just unpublished below, and again, we do see exactly the same site. We see this pie chart. I really like those that PEAK software provide to you, not only what sugars was detected, but what the percentage of these sugars is detected, sometimes to very low abundance. I tried myself actually go back to the spectra and find it, and with software, it's very hard. That's why it's easier to go to peak straight. Don't even bother with the raw spectra. But that's very much important to show actually what the main sugars are and what those that are really in the background. Another area on the top of the pie chart of the unpublished, on the sites that actually been found, you can see on the top, they showed as a bar the 3 different types of IgAs that we've been looking at, basically native, glycosylated, and acylated. And that's, again, very, very clear cut off where and which sugars are very important for, in our case scenario, for the peanut allergy, and those that are not being perturbed compared to the native state. That's kind of very nice view, at least for me, to look into where the areas are most important for our study. Another view, again, pie chart of what is there, as well as another representation. I really love this representation. It tells you how complex this thing is. I think that will be my favorite slide when I will go to the conference where the people will start talking about RNA sequencing. I usually would show this particular slide and tell them that good luck with RNA sequencing. This is just single protein and single site, and that's how the complex this thing is. And as well as it just immediately just picks you which exactly particular sugar types you would like to look at as a most possible, drug target. As well here, totally another side. Again, representation of the sugars. Those are changed, those are not changed. And again, we're still speaking about one protein. There's no other things. I have not switched. This is just 3 samples being done each in 30 minutes of mass spec run. That's it. That's how much data we have now from this particular case. Here's the little bit of the coverage where I could just I was telling you before, where there is a certain region which is very important. It's been actually known to be very important of the changes on the glycosylation part on this particular region are kind of quite difficult to catch. And that's how the area where we stuck, just to see on the hinge region how we can very good coverage. As you can see to the left and to the right, that was as well the Albert was noting, right, depth of the coverage we have right there versus the hinge, which just kind of have only one peptide. That's where the importance was, just to make sure we see this particular peptide that comes actually from the lysine digestions, to make sure we understand what that sugar pattern is on that particular region. Again, even on that region, we can have a comparison, what is changing, what is not, compare from the native to the peanut butter allergy process, and how we can address this towards the possible treatment. Again, this hint region gives us 2 areas where it wasn't actually noticed before, at least what from our knowledge. And then this will be our main message to our collaborators in Massachusetts General Hospital, where this project we're working with together, that they might need to pay attention to these particular sites of the glycosylation as a new target for drug treatments. And the last one, thank you very much for the bioinformatics solution support. Thank you, Christina, for all your work. It's really amazing work. And of course, I would like to thank all the people on my biomarker discovery and drug laboratory. And thank you for your attention.